Good day, class, and welcome to our lesson on glycolysis. This will be our lesson outline. Let's begin with an overview of glucose metabolism. Glucose is the transported form, while glycogen is the stored form. Now direct your attention to the image in the right. You have here illustrated the intestines, the liver, and the extrahepatic tissues or tissues other than the liver. You will see in this image that the diet is digested and absorbed in the intestine, and the final products of digestion, which are then absorbed, are the simple sugars, fructose, galactose, and glucose. These then feed into the liver, where fructose and galactose are converted to glucose and stored as glycogen, and glucose, furthermore, as itself is stored as glycogen. When the body has low levels of glucose after a fast or a period of not eating, the stored glycogen in the liver is released into the plasma as glucose. You will also see here that the extrahepatic tissues liberate amino acids, lactate, and glycerol, which then goes through the circulation and enters the liver and is converted by the liver through the process known as gluconeogenesis into glucose. Right from this diagram, you will understand that all the cells of the body will utilize glucose, and therefore glucose has to be transported by the circulation, and that glycogen is some form of a buffer for the levels of glucose. It is the storage form of glucose. Glucose uptake into the cells is regulated. As you understand, this is through the action of insulin and non-insulin dependent glucose transporters. In particular, insulin-dependent GLUT4 uptake decreases during a fast because when you don't have something to eat, the levels of insulin go down, and therefore the activity of GLUT4 goes down. This in effect redirects glucose from uptake by muscle and fat to uptake by glucose-dependent structures or cells such as found in the brain and the erythrocytes. So, in essence, when you don't have something to eat, the low levels of insulin prevent uptake by muscle and fat and thereby spare the glucose for more important cells such as the brain and cells that cannot utilize other forms of energy such as the erythrocytes. All cells of the body are capable of glycolysis, which starts in the cytosol and ends in the mitochondrion. What is the goal of glycolysis? Glycolysis in general is a form of glucose oxidation. That means glucose is the source of electrons, and through glycolysis, electrons are removed from glucose. It turns one molecule of glucose, which is our reactant, into two molecules of pyruvate, which are the products of glycolysis. Glucose is a 6-carbon sugar, and therefore, upon splitting of glucose, you will form two molecules of pyruvate, which are 3-carbon sugars. Pyruvate is then transported from the cytosol into the mitochondrion, where it is transformed into acetyl coenzyme A. And acetyl coenzyme A is taken up by the Krebs cycle or the TCA cycle, otherwise known as the tricarboxylic acid cycle, where reducing equivalents, which technically just holds electrons, and these are your reduced NAD or NADH and reduced FAD or FADH2. These byproducts or reducing equivalents then go into the inner mitochondrial membrane and matrix, where electrons that they hold are reoxidized in the respiratory chain, otherwise known as the electron transport chain, to generate high amounts of ATP. So in the image in the right is a summary. Glucose, through glycolysis, produces 2-pyruvate, which at the same time produces 2 molecules of ATP and 2 molecules of NADH. That NADH can then directly go into the electron transport chain. The pyruvate undergoes an intermediate step, which we will discuss later, and that is catalyzed by pyruvate dehydrogenase, which in essence removes 2 carbon dioxide molecules to produce acetyl coenzyme A and again NADH. Acetyl coenzyme A is then taken up by the Krebs cycle, which produces massive amounts of the reducing equivalent NADH and FADH2, and again produces substrate level phosphorylation GTP. These reducing equivalents all together are reoxidized in the electron transport chain, where oxygen takes up the electrons and is converted to water, but at the same time, ATP is produced. So let's now go through the steps of glycolysis. Glycolysis is initiated, it is started by phosphorylation. Phosphorylation means the attachment of a phosphate group into a molecule, and this molecule here in focus is glucose. Glucose that has entered the cell by means of GLUT or glucose transporters is phosphorylated by hexokinase. Therefore, hexokinase is the enzyme that attaches phosphate groups to glucose. And the donor of the phosphate group would be none other than adenosine triphosphate. So you will see that in the upper portion of the image, where a glucose molecule with that structure receives a phosphate group that is attached to carbon number 6. And therefore, the product is glucose 6-phosphate. Glucose 6-phosphate is important because it is highly energetic and therefore reaches the required activation energy for glycolysis. It is highly energetic and it's negatively charged. Its charge is important because it serves as a trapping mechanism. After glucose is phosphorylated, it has a harder time leaving the cell, and therefore, once trapped inside the cell, it has to undergo metabolism. Glucose 6-phosphate then is converted to fructose 6-phosphate, which is a functional isomer of glucose. This is catalyzed by the enzyme phosphoglucose isomerase. You have to understand that in the image that is a double-sided arrow, which means that the levels inside the cell of glucose 6-phosphate and fructose 6-phosphate are equal. They are in equilibrium. The second phosphorylation is catalyzed by phosphofructokinase 1. And again, the donor is adenosine triphosphate. And therefore, at the end of the reaction, you will have 2 phosphate molecules. One attached to carbon number 1 of fructose, and the second one attached to carbon number 6 of fructose. This second phosphorylation by phosphofructokinase 1 is the committed step in glycolysis, which means that after fructose 6-phosphate is phosphorylated, that molecule of originally glucose can no longer go into other metabolic pathways. It will have to be catalyzed and taken up by glycolysis and no other accessory pathway. So that's what the committed step means. The next step would be basically hydrolysis. Aldolase cleaves fructose biphosphate into 3-carbon products. So fructose 1,6-bisphosphate, which is a 6-carbon sugar, highly energetic because of the 2 phosphate groups, is hydrolyzed by aldolase to form 2 molecules of 3-carbon sugars. And these 3-carbon sugars include dihydroxyacetone phosphate, which is a ketone, and glyceraldehyde 3-phosphate, which is an aldehyde. You have to look at the arrow again. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are represented by a bidirectional arrow, which means that they are also in equilibrium and they can interconvert with one another. And that is through the enzyme triose phosphate isomerase. In the zoom in photo, we will understand that only glyceraldehyde 3-phosphate proceeds into the further steps of the reaction. So glyceraldehyde 3-phosphate is taken up by the next phase, and DHAP or dihydroxyacetone phosphate would have to be converted to glyceraldehyde 3-phosphate to undergo metabolism. The next step would be the oxidation of glyceraldehyde 3-phosphate. And this is via the reaction catalyzed by a dehydrogenase enzyme. And that dehydrogenase enzyme, by its name, removes a hydrogen group and transfers that hydrogen group to NAD, forming reduced NAD or NADH. Because of that removal of hydrogen, there will be a highly energetic bond formed. And that is represented here by 1,3-bisphosphoglycerate. By removing hydrogen, you form a highly energetic bond that is unstable, which is stabilized by the attachment of another phosphate group. Therefore, from a sugar, 3-carbon sugar with only one phosphate group, you end up with 1,3-bisphosphoglycerate, a triose with 2 phosphate groups. The next step would be the simple hydrolysis of bisphosphoglycerate. So that 1,3-bisphosphoglycerate, the phosphate group there is hydrolyzed, and that releases massive amounts of energy. And so the body will not waste that released energy, and that energy is captured into adenosine triphosphate through substrate level phosphorylation. And that forms 3-phosphoglycerate and a molecule of ATP. That 3-phosphoglycerate has a phosphate group at position number 3. You will understand that this is not geometrically stable. And so what the body does is transfer the phosphate group into carbon number 2 through the action of phosphoglycerate mutase. A mutase enzyme transfers functional groups from one position to another. Here, from position number 3 to position number 2. And so our product would be 2-phosphoglycerate. 2-phosphoglycerate will then be dehydrated. You remove water molecule to form phosphoenol pyruvate, and therefore the formation of a double bond. This phosphoenol pyruvate has a free phosphate group in the middle, and therefore this free phosphate group will be taken up again by adenosine diphosphate, releasing lots of energy and captured into adenosine triphosphate, another substrate level phosphorylation, forming pyruvate. But as you will surmise, after the step of hydrolysis of fructose 1,6-bisphosphate by aldolase, you will have two sugars. And therefore, each step that we have discussed in the 3-carbon sugar step would be multiplied by 2 because you have two molecules entering the pathway. Here illustrated is the intermediate step by which pyruvate is decarboxylated in the mitochondria into acetyl coenzyme A. This is catalyzed by an enzyme complex, which means that this enzyme has 3 functioning parts, which is greatly illustrated here from the image from Harper's. So you have enzyme complex, enzyme 1, 2, and 3. Enzyme 1 is pyruvate dehydrogenase. Enzyme 2 is dihydrolipoyl transacetylase. And enzyme 3 is dihydrolipoyl dehydrogenase. So let's first look closely into the images. You have the starting molecule or reactant pyruvate. By the action of thiamine diphosphate TDP, that pyruvate is converted into hydroxyethyl. Hydroxyethyl, by removal of carbon dioxide from pyruvate, will then be acted upon by lipoic acid. So for enzyme number 2, let's start at the 6 o'clock position, where you have dihydrolipoamide. This dihydrolipoamide has 2 free sulfhydryl groups. For you to form the highly energetic oxidized lipoamide, you need to connect the 2 free sulfhydryl groups. And that is through an oxidation reaction that is catalyzed by enzyme 3. So FAD will take up the hydrogen from the 2 sulfhydryl groups, SH groups, of dihydrolipoamide, forming a closed ring structure where sulfur is connected by a covalent bond with the next sulfur. This highly energetic compound will then be able to take up the hydroxyethyl from pyruvate dehydrogenase, where that is represented in position 11 o'clock of enzyme 2. This is highly energetic, and therefore, for the body to stabilize the molecule's energy, it will have to attach coenzyme A. Coenzyme A will then bind with the hydroxyethyl moiety, and that releases acetyl coenzyme A. You will understand from this image that there are lots of reacting molecules, but clinically, these reactants are derived from vitamin B1, thiamine, that's your TDP, vitamin B5 or pantothenic acid, that is where you derive coenzyme A, vitamin B2 or riboflavin, and that is where you derive FAD or FAD, vitamin B3 or niacin, that's where you derive NAD, and lipoic acid, which is utilized in enzyme number 2. So this is quite heavy. Let's summarize. Pyruvate is taken up by pyruvate dehydrogenase, and the end product there would be hydroxyethyl after releasing carbon dioxide. Hydroxyethyl is taken up by enzyme complex number 2. It binds first to lipoic acid, which serves as somewhat like a plate that holds hydroxyethyl as if hydroxyethyl is the food. And you attach coenzyme A, which is much like the sauce, to the food, which is hydroxyethyl. Therefore, release acetyl coenzyme A, which is the food you take into your mouth. So that's a simple analogy. For you to produce lipoic acid, highly energized form, you need enzyme complex number 3, which will form the closed ring structure via oxidation, and that utilizes fat. And so at the end of the day, you start with pyruvate and you end up with acetyl coenzyme A. Now let's go through regulation of glycolysis. Glycolysis is a very important reaction, as you will surmise, because it occurs in all cells and it's how the cells utilize glucose. But as you will also observe, there are a lot of steps in glycolysis. And the reason for that is you don't want a reaction to produce your end products in only just one go, because that would mean that the release of energy would be occurring in only one step. And so the energy released in one step would be very high. That would mean loss of lots of heat energy. So if glycolysis would occur in only one step, then that would mean you would burn up. And so what the body does is chop the steps into smaller, multiple steps so that there is slower release of energy and staged release of energy so that the body has a time to capture that energy into ATP. And so let's proceed with discussion of how glycolysis, which is very important, is regulated. The first form of regulation is through the molecule fructose 2,6-bisphosphate. Here you have to differentiate fructose 1,6-bisphosphate, which has phosphates at position number 1 and 6, from fructose 2,6-bisphosphate, which has position of phosphate at carbons 2 and 6. Fructose 1,6-bisphosphate is a reactant of glycolysis, whereas fructose 2,6-bisphosphate is only a regulator of glycolysis. So during periods of high glucose loads, the level of fructose 6-phosphate will increase. This will then be fed into fructose phosphate, phosphofructokinase 2. So the high levels of glucose will produce high levels of fructose 6-phosphate, which will saturate phosphofructokinase 1, the enzyme in glycolysis. And the rest of the fructose 6-phosphate will then spill into phosphofructokinase 2, another enzyme. That phosphofructokinase 2 will then produce fructose 2,6-bisphosphate. So again, fructose 6-phosphate is converted into fructose 2,6-bisphosphate. How? By high levels of fructose 6-phosphate and by the activity of insulin. You have to understand that after you feed and you have digested and absorbed the food, the levels of insulin will go up. Okay? And it has lots of effects. And one of its effects would be to facilitate the conversion of fructose 6-phosphate to fructose 2,6-bisphosphate. High levels of fructose 2,6-bisphosphate will then stimulate PFK1, which is the enzyme you find in glycolysis. High activity of PFK1, which is the committed step, will then speed up glycolysis. So let's recapitulate. High glucose load, high levels of insulin. High glucose load, high levels of fructose 6-phosphate through glycolysis. High levels of fructose 6-phosphate will saturate a lot of the PFK1, phosphofructokinase 1, and the rest will then be taken up by PFK2, which will then produce fructose 2,6-bisphosphate. The production of fructose 2,6-bisphosphate from PFK2 is further stimulated by insulin. The product of PFK2, which is fructose 2,6-bisphosphate, will then produce positive regulation or stimulation of PFK1, which is the committed enzyme or step in glycolysis, which causes glycolysis to speed up. Therefore, in summary, high glucose load, speeding up of glycolysis. Lastly, you have another point of regulation, which we have touched in anatomy and physiology, and that is through glucokinase, which is an isoenzyme of hexokinase. Glucokinase, like hexokinase, catalyzes the attachment of phosphate groups in glucose. But glucokinase is specifically found in the liver and the pancreatic beta cells, which secrete insulin. As you will observe in this image in the right, hexokinase is illustrated with the red curve, and glucokinase is illustrated with the blue curve. On the x-axis, you have glucose concentration, and on the y-axis, you have the rate of reaction, which basically tells you the state of activity of the enzyme. Hexokinase, the red curve, is active. It has high Vmax or high y-intercept even at low glucose concentration. Therefore, even at low glucose concentration, hexokinase is active. While glucokinase, which is illustrated using the blue curve, will only achieve a high reaction rate when the glucose concentration is high, which means it will only be active after you have taken a meal, digested the meal, and absorbed the carbohydrate. That's the only time glucokinase will be activated. And therefore, we can say that glucokinase has a high Michaelis constant. It will need a lot of glucose to be active. That's what a high Michaelis constant means. Therefore, it is active only during higher plasma glucose concentrations after a meal, and therefore it acts as a glucose sensor in the pancreas. You have to understand that the response of the beta cell of the pancreas would be to release insulin when there are high levels of glucose in the blood. And that is achieved by the action of glucokinase, which is active only at high levels of glucose. Therefore, it can act as a glucose sensor. In the liver, glucokinase acts as a glucose buffer. So, whenever you have taken a meal and the portal blood has high levels of glucose, this will mean that in the liver cells, glucokinase will be active. By attaching phosphate groups to glucose, it will create glucose 6-phosphate inside the liver cells, which is highly energetic and negatively charged and traps the glucose within the liver cell, so that the liver cell acts as a mesh. It acts as a captor of glucose, so that after a meal, you will not have erratically high glucose levels in the blood. Okay? And so, through this lesson, we have discussed glycolysis, how it is performed, how it is regulated, and how it is essential to all cells of the body. Glycolysis starts with glucose and ends with pyruvate, which is further converted into acetyl coenzyme A through an intermediate step that occurs in the mitochondrion. That intermediate step utilizes the enzyme complex pyruvate dehydrogenase, which is highly dependent on a lot of B vitamins. Thank you for listening, and I hope you learned a lot.